machine learning is very experimental. So this
Hmm.
is your toolbox. You have these techniques that you can try, and you're going to have to tweak each of them because there's different parameters and intensities related to each of them that you need to adjust. So the best solution tends to be try some combination with it within this toolbox, tweak it as needed for your particular task and for your particular data.
So basically, the model is giving you feedback on what's working best.
Learn about the newest digital pathology trends in science and industry, meet the most interesting people in the niche, and gain insights relevant to your own projects. Here is where pathology meets computer science. You are listening to the Digital Pathology Podcast with your host, Dr. Alexandra Zhurov.
I have Heather Couture as my guest today, the founder of Pixel Ciencia, and I came across this article of Heather's about 5 ways to make histopathology image models more robust to domain shifts. The supervised deep learning has basically taken over or has put itself everywhere in the image analysis approaches for pathology, but this is a very data-centric approach, and data-centric in this context means that we train our image analysis solutions on data with certain appearances, and then we want it to perform well on similar data. But how similar this data has to be? How similar do the whole slide images we are working with have to be? Is it enough if they come from the same lab, or do they have to be stained in the same batch and scanned with the same scanners? And what about whole slide images from different labs, different countries, or from different times if we do archived slides? So I have Heather as my guest. Again, she's the author of this article. Welcome, Heather. How are you today?
Good, thanks. Thanks for having me.
So let's start with, what is a robust solution in supervised deep learning, or basically in deep learning for pathology? And let's talk about, why did you write this article?
Robust can mean different things in different contexts. And in this case, and specifically with pathology images, it's meaning robust to changes in the images. So it could be they were scanned on a different scanner, they're from a different lab, the stain intensities are different because of something in the processing, or they've faded over time, or maybe a different population of patients. So any of these changes can create a difference in appearance in your images. And if you're trying to apply a model that was trained in a source domain, let's say from one scanner, and you're trying to apply it to a target domain, let's say a different scanner, something about those images may be different that the model doesn't know how to handle. So maybe your training set of images were from the Apereo scanner, and you have another data set of images from Hamamatsu, and you want to apply your model there. But if those target images look different than the source ones, the model can fail, and in particular, it can fail in unexpected ways. So the challenge is that we want to create a model that will also perform well on the Hamamatsu scanner in this setup, or perhaps even other scanners. And similar challenges occur when the tissue is stained differently, it's faded over time, or something else changes. Maybe you have a model for nuclei segmentation for one type of cancer, and you want to apply it to a different type of cancer. That's also a domain shift.
So basically, domain is like a group of similar images, similar data points. Like you said, domain would be all images scanned with the Apereo scanner, and another domain would be scanned with Hamamatsu. Do I understand
Right.
it correctly? Okay.
Right. And domain could be with respect to scanners. It could be with respect to the lab or the
Mm-hmm.
location in the world, or any other kind of grouping like that.
And domain shift would be, we want to apply solution from one domain on a different domain that is similar, right? Similar meaning we are still working on H&E stained whole slide images.
Right. But different in that, different scanner, different lab, anything like that. So it's some sort of target domain that's different from the source.
So if there was no option to account for this domain shift, to account for the dissimilarities in those similar images, how similar would the samples have to be if there was no option to account for anything?
It's hard to quantify that, and that for a model to perform well on test images, those test images need to be from the same distribution as the training images. And so if they look quite similar, it's hard to quantify the term look, whether they appear similar. If they are similar enough, it'll perform well. If they're different enough, the model will fail in unexpected ways. And especially with deep learning models that have a lot of moving parts, a lot of parameters when you have to train these models, if something changes in the image, if it's something that changes in your data set over time, or you've gathered images from a different place, that can be enough for the model to fail. What needs to happen for the model to work is the images need to look similar enough to something that the model has seen when it was training. And so the features that model learns, it needs to be able to characterize those same features with your target images.
And usually it has to be more similar than it would be for a human observer, right?
Definitely for a human observer, but there can also be other subtleties that a machine learning model could pick up on that a human observer could not.
Yeah, so for example, I don't care if the slide is scanned on Apiri or Hamamatsu or whatever, I'm going to approach it the same way. But this is following with our example, this would be enough for a model not to perform well.
It could be, and then I can't say it will always fail in that scenario, but it could fail.
So before I read your article, and I will link to it in the show notes, I knew about two approaches. The first approach was to train on not so similar images, so across different domains to account for those variabilities, or do the stain normalization with GANs, generative adversarial networks. But from your article, I learned that there are several more approaches that can help us. And I thought with those two, I was already very on top of the matter. Apparently not. So how many solutions do we have, and what are the ways to increase the robustness to domain shift of histopathology models?
Well, you're definitely ahead of the game by understanding that there's a problem and that there are some solutions. But there's 5 different ones that I'm aware of that I wrote about in this article. One is stain normalization, like you said. So the goal is to make the target domain images look similar to the source. So in the case of H&E, make the hematoxylin and eosin stains look similar between the target and source domains. The next is color augmentation, and this is in particular used in deep learning models. The goal of that is to increase the diversity in the source domains to teach the model how to handle different situations. So to make it more like
robust. The opposite of the stain normalization.
Right. The third one is adversarial domain adaptation. And this is to encourage the model to learn features that are more domain invariant. So the key here is the adversarial. So you're going to train a model to do the task that you're trying to do, but you're also going to try and teach it not to be able to predict which domain the image came from. So you're going to train it with your labeled training set from the source domain and also some unlabeled images from the target domain. And you're going to ask it to predict the domain and adversarially train it so that ideally it will not be able to predict that domain.
So that it doesn't focus on the things that are specific for the domain.
Right. The fourth one is to adapt the model for the target domain, and this isn't possible with all models, but some models incorporate statistics like the mean and standard deviation of features within the model. If those statistics have changed from your source to your target, you could recompute them on the target domain, and from that have your model perform better on the target domain. So for that, you need unlabeled images from the target domain. And the last one, which tends to be the best solution, but only if you have the data, because this does require labeled images from your target domain, is you initially train your model on the source domain, but then you do what's called fine tuning on the target domain. So essentially you continue training it a little bit more on your target domain so that it can learn any unique characteristics of the target domain.
But then you have a constantly evolving model.
Right. Where you will need to fine tune it or train it more for each target domain that you're trying to apply to. Yes.
Can all those 5 methods be used simultaneously, or it does not work like that? And if it doesn't, which would you recommend, or how does it work?
Not necessarily all 5 together, but definitely combinations within that in that subset of 5. It's going to depend what you have available. If you have enough labeled images from your target data set, fine-tuning your model on that target data set is going to give you the best result. But you don't always have labeled imagery from your target data set. Sometimes you don't have any images from your target data set, or sometimes they're only unlabeled because the goal of the model is, of course, to predict whatever you're trying to predict on that target domain. So in that case, if you have unlabeled images from your target domain, you could take the model adaptation approach to recompute some of the statistics, and that's only possible with some models. Other than that, it tends to be a combination of the other 3. So adversarial domain adaptation, color augmentation, and stain normalization. And each of these have different goals, but when you bring them together into a single model, or sometimes it's subsets of two of those, that tends to, from what I've seen, give the best results. But it's not necessarily these 2 are going to always give you the best result within that. Machine learning is very experimental. So this is your toolbox. You have these techniques that you can try, and you're going to have to tweak each of them because there's different parameters and intensities related to each of them that you need to adjust. So the best solution tends to be try some combination within this toolbox, tweak it as needed for your particular task and for your particular data.
So basically the model is giving you feedback on what's working best. So I understand if you are designing your model from scratch or programming it in a programming language, you're familiar with those approaches, you can do it. Question here is, can these methods also be used when the models are being developed with commercially available software by non-computer scientists? Or is it something that has to be incorporated into the software and this is user independent?
In most cases, it would need to be incorporated into the software, and I'm not familiar with what's in each individual commercial software package. Some of these require modifications to the model itself, in particular adversarial domain adaptation. Some of them, like color augmentation, is done during training, and it's very common to use already. You just might apply it slightly differently or with a greater intensity if you need to improve your robustness to domain shift. So some of those might already be incorporated. Stain normalization, you could perhaps apply outside a software package. Take the images that have been scanned, apply the normalization to make your target images look more like your source domain, and then put it through whatever toolkit you're using to train and to apply your model. In the case of fine tuning, that requires first training a model on your source domain and then tuning it some more on your target domain. Do that. Some software package will allow that.
Yeah, I think this is something that you can just do with whatever you're working if you're not a computer scientist. With each of the software packages that I'm aware of, you just take the new data and let it run on it and adjust to the extent it's needed.
Yeah.
So you would recommend that the options, or at least some of them, are incorporated into a software package?
Yeah, without any of these options, models definitely do fail on new domains, and maybe not all domains, but some domains that you might want to apply your model to. And so to improve the robustness to that, some of these tools, not always all of them, but some of them do need to be incorporated to improve the robustness of models.
Do you have an example of a model you worked on that failed miserably because you were not aware of the domain shift, or the domain shift was so drastic but not really visible for a human observer? Do you have any example of?
I don't think I have an example of a drastic one. There's one data set I was working with during my PhD. I think the images were all scanned on the same scanner, but they were archival slides. So some of them, the stain had faded over time, and some of them had been in storage longer than others. And so for that one, it wasn't a specific domain A and domain B. It was these images had different amounts of fading. And so for that case, we did apply stain normalization to get the stain intensities in a more similar realm before modeling.
Thank you very much. This is very informative. I think many people are aware that it doesn't work across the board, but I don't think to that extent to pay attention to like those little things that a pathologist or a human observer just totally sees through, like faded stain because you took slides from the archive. Thanks so much for explaining this to us.
Oh, thanks for having me.
And you wrote more articles like that, so tell the listeners where they can find them.
My website, which is pixelscientia.com, pixelscientia.com.
Okay, I'm gonna leave this link in the show notes, and I'm gonna also link to our previous podcast episode. Thank you very much and have a great day.
You too. Take care.
Thanks for listening. For more great digital pathology resources, visit the Digital Pathology Place website and subscribe to our newsletter on digitalpathologyplace.com. After subscribing, you will get access to the free digital pathology crash course, which will help you start working on your digital pathology projects immediately. Talk to you in the next episode.